All right, Ninja Nerds, in this video we're going to talk about gustation. So what is gustation? Gustation is basically the sense of taste. Now, why is that really important? Because, I want to taste my food. I know you guys do too. So let's go ahead and talk about how this taste is actually occurring. Before we do that, let's look at a little bit of anatomy of the tongue. All right, so if we look here, you're going to have the tongue, right? This is the anterior part of the tongue. And this is going to be the posterior aspect of the tongue. You can tell that because, again, this would be the mandible. Just a crude diagram, and again, this would be the nose, the upper part of the maxilla, mandible. And this is going to be the trachea, larynx with the epiglottis, right? All right. So if you're looking at the tongue, there's going to be specialized things called taste buds. Okay, so you're going to have these things called taste buds. And taste buds are kind of scattered throughout the entire tongue. I'm going to draw them in like this little swirly thing right here, right? So this taste bud that's found more in the anterior 2/3 of the tongue, right? That I'm drawing here, these swirly ones. This is called the fungiform papilla. This is called the fungiform papilla. And the fungiform papilla is found throughout the entire tongue. It's just a little bit more concentrated in the anterior 2/3 of the tongue. Now, how am I telling you where the anterior 2/3 of the tongue is? All right, so here's the tongue, right? If we look in the back here, in this back part, you see this little V part right there? That V is a specialized structure, and they call that the sulcus terminalis. And the sulcus terminalis is what basically separates the anterior 2/3 of the tongue from the posterior 1/3 of the tongue. So anything in front of the sulcus terminalis is going to be what? Anterior 2/3. Anything in the back of the tongue is going to be posterior 1/3. Okay. All right, sweet deal. So now, there's going to be more of these fungiform papilla. They're just not going to be as concentrated within the posterior 1/3. Then there's going to be some other ones. There's some other papilla that are actually located right here in this V-like structure here. On this V-like structure is going to be what's called the circumvallate or the vallate papilla. Okay, so these are called the circumvallate or the vallate papilla. So, okay, what are these black ones here called? These black ones here are called the fungiform papilla. And then these maroon ones here are going to be what's called the circumvallate papilla. Alrighty, so that's the circumvallate papilla. There is other ones too, not as important. They're actually going to be a little bit more important during the younger times in life, and they're actually going to be on the sides of the tongue. They're going to be on the sides of the tongue. And these ones on the sides of the tongue are actually called the foliate papilla. This is actually called the foliate papilla. And they believe that this one might have a little bit more of a function specifically with the taste of milk in younger children, right? But again, they're not completely sure about that, but they do believe that it has some type of relationship with the taste of milky-like substances. All right? There is another one, but the other one is actually not going to have any type of taste buds in them. It actually is going to have, it's kind of arranged in like rows, and it's conical in shape. And basically, what it's designed to do is to provide a friction surface, so a surface that has a lot of friction. So that, whenever a perfect example of this one is whenever you're eating ice cream. When you're eating ice cream, you have to use your tongue. And what these little conical ones that are aligned in rows are doing is they're helping to develop some friction on the actual tongue surface and the ice cream to lick the ice cream off. What is this friction-like surface, that papilla called? They're conical, they're aligned in rows, but they do not contain taste buds. This is called the filiform papilla. All right, cool. So we went over the filiform papilla, which is more for like the friction. It's conical, arranged in rows, and it's more for being able to lick off things like ice cream. Like that's an example. Fungiform papilla is more concentrated in the anterior 2/3, but it's also found in the posterior 1/3. And they're going to contain taste buds. Foliate papilla is found on the sides of the tongue, all right? But they're not as functional during the adult years. They're more functional within the younger years, they believe, and with more specifically with the taste of milk-like substances. And then in the right here on that sulcus terminalis part here, you're going to have the circumvallate or the vallate papilla, right? And they're going to contain taste buds. There is one more structure. You know here, the epiglottis? The epiglottis is also going to consist of a little bit of taste buds. So here's your epiglottis. And the epiglottis does have a little bit of taste buds. You know what else has taste buds? You know the part of the pharynx? Let's say that I have here nasopharynx. Let's say here I have oropharynx. And let's say here I have laryngopharynx. Okay? Some parts of the pharynx might also have a little bit of taste buds, primarily that of the oro and a little bit of the laryngopharynx, right? Okay. So, we got taste buds in the actual, a little bit in the pharynx, a little bit in the epiglottis, the circumvallate papilla, the fungiform papilla, the foliate papilla, and the filiform papilla. Great. All right. Now what I want to do is I want to zoom in on this taste bud. Because in a taste bud, so like for example, let's say I took this taste bud right here, and I look at it like this. So let's say here, I had it like that, right? Let's say here's the taste bud. In the taste bud, it consists of multiple taste cells. So for example, there could be 3 types of cells. Type 1 cell in here. You could have another one, which is going to be like a type 2 cell. And you could have another taste cell, which is going to be type 3 cell. We're not going to spend a lot of time about these. We are going to talk about the different types of taste. All right, so this is a taste bud. This whole thing housing them is called a taste bud. But in the taste bud, you're going to have taste cells. And then these taste cells are actually going to be connected. They're going to have connections to dendritic processes of certain nerves. So they're going to be able to have dendritic processes that are connected with certain nerves. So they have dendritic processes, terminal connections. So they're going to have little terminal connections here that are going to be connected with the taste cells. So you're going to have little synaptic portions here that are going to interact with these taste cells and pick up certain types of taste from each one of these cells and carry it on certain cranial nerves, and we'll talk about those. All right, cool. Let's zoom in on these taste cells. Well, we're going to make it just one big taste cell, because one taste cell can actually consist of multiple, one taste cell can have multiple receptors for each type of taste. Before we go into all the actual transduction mechanism of a taste cell, what are the different types of taste? Because we talked about olfaction, right? And how olfaction is going to be consisting of many different types of odors. Well, they weren't able to package olfaction into different separate types of odors. Where taste is a little bit more specific. We were able to package it into special types of categories. So for example, we could say taste could either be sweet. Okay, so for example, if it's sweet, think about certain substances that are going to be sweet. They're going to contain a lot of glucose or sucrose or different types of polysaccharides or monosaccharides or disaccharides, right? So think about that. Think about chocolate. I don't know, that's the first thing you think of, right? Chocolate. Okay. What about maybe, what about, let's say, salty? So then there's a second one, which is going to be salty. So salty, you know that this is going to be rich in primarily sodium chloride. So it's going to be rich in sodium chloride. What about the taste like sour? What if something is sour? Well, if it's sour, it's rich in protons. Or if it's sour and it's rich in protons, it's more acidic. So if it's more acidic, think about certain types of acidic foods, like fruits. So fruits is a good example of certain types of sour substances. What about bitter? Bitter is going to be kind of like something that's rich in a chemical, specific types of chemicals, like one of them could be quinine, could be things like, caffeine, so you're drinking your coffee and stuff like that, could be nicotine, a lot of different chemicals, right? But bitter is usually going to be consisting of certain types of alkaloid substances. You know another one that's non-alkaloid? Aspirin. Aspirin's another one too. Aspirin actually has a bitter-like taste. And one more, and this one is a funny one, and this is called umami. Umami. And this is going to be really, really rich in a certain type of chemical, a certain type of amino acid called glutamate. Monosodium glutamate. And other different types of amino acids that's in the L type of position, so L amino acids. Certain types, okay, but primarily of the most important one is glutamate. And so this is going to be rich in a lot of beef-like substances. So certain types that, you bite into a big old juicy cheeseburger. Umami, right? That's going to be rich in glutamate. All right, so now we have all of these different types of categories of taste. So these are my taste categories. All right, and again, it's sweet, salty, sour, bitter, umami. And then again, sweet, I said, would be consisting of a lot of different types of, usually some type of polysaccharide or monosaccharide. All right, cool. Now, let's see how these different substances activate a taste cell. All right? Now, let's start with the first one, which we're going to go down in an order, sweet. All right, sweet is actually going to act through what's called G protein coupled receptors. This one acts through a G protein coupled receptor. All right, so let's say for whatever reason I have a Hershey's chocolate bar. All right, and this Hershey's chocolate bar is going to be rich in a lot of glucose. So let's say here's my glucose molecule. And what happens is my glucose molecule comes over here and he binds onto a specific type of receptor. Let's say here I put my receptor. Here's my G protein coupled receptor. And this G protein coupled receptor is bound to a protein called transduction. So it's bound to a G protein called transduction. And whenever glucose binds onto this receptor, he activates, so come over here, he'll bind onto this G protein coupled receptor, he'll activate transduction. What transduction will do is going to come over here, and it's going to activate a special effector enzyme that's located within the cell membrane. Look at this guy right here. This guy here is going to be called phospholipase C. And phospholipase C is really special because when transduction binds into phospholipase C, he activates phospholipase C. And what phospholipase C does is he takes a special type of chemical that's embedded inside of the membrane here. And that chemical is called PIP2, phosphoninositol diphosphate. And what phospholipase C does is he breaks him into two different components. One of the components is called diacylglycerol. And diacylglycerol will actually go and activate an enzyme called protein kinase C, who will go and phosphorylate certain types of channels. So he might go and phosphorylate specific types of protein channels. We'll talk about that in a second. Okay, so we could put phosphate ions on certain types of protein channels. But of the more powerful effect is going to be the other molecule. The other molecule is actually going to be what's called IP3, which is inositol triphosphate. DAG is diacylglycerol. IP3 will come to a specific part inside of the cell. And this cell is going to actually have an organelle here. Let's say I draw the organelle like this. And this organelle is going to be called the smooth ER, or the endoplasmic reticulum. IP3 has special receptors that are present on the smooth endoplasmic reticulum. And what happens is he activates these channels that are present on the smooth ER. When he activates these channels, look, IP3 will come over here, and it'll activate this IP3 receptor on this channel. When he activates this channel, there's going to be a lot of calcium ions inside of this actual smooth endoplasmic reticulum. And what's going to happen is it's going to start pushing calcium ions out. As the calcium ions start becoming very, very in high concentration inside the cell, what is this going to do? I'm glad you asked. You know what else is really cool about these taste cells? They have a lot of different types of vesicles here. So they have these vesicles here. And these vesicles are rich in certain types of neurotransmitters. You know what kind of neurotransmitters that are usually consisting of? They could be consisting of ATP, or they could be consisting of a chemical called serotonin, which is another different type of neurotransmitter. What happens is calcium will come over here. You know there's certain types of synaptoproteins here? So here could be a protein here could be a protein here on the membrane of this taste cell. And then look what happens here. There could be another protein right here, and another protein right here on these synaptic vesicles. What calcium does is, calcium will come over here, and the calcium will help to activate this process. So it'll help to stimulate these two synaptoproteins to interact. When these synaptoproteins interact, look what happens. The actual synaptic vesicle membrane and the actual cell membrane fuse together, and they release out the chemicals onto what? Well, what was in this vicinity? What did I show you before? Remember, there was going to be a nerve terminal right here, right? So here could be a nerve terminal of a specific type of cranial nerve. And we'll talk about those in just a second after we go over this pathway. So right here could be a nerve terminal. And when you release this actual neurotransmitters, these neurotransmitters can go and can stimulate this nerve terminal and generate action potentials down this actual nerve. Okay? Cool, that's that guy. Now, of the lesser effect, you could have protein kinase C over here. Protein kinase C could phosphorylate certain types of channels, and it could phosphorylate channels that could actually either cause potassium ions to not leave the cell, or it could cause calcium ions to come into the cell, right? It could be a lot of different things, but this is usually of the less effect. Not going to have a very potent effect. Now, guess what's nice? Sweet substances perform this pathway. You know what else performs this pathway? Umami. So of these, which ones perform the G protein coupled receptor pathway? Umami performs the G protein coupled receptor pathway. Bitter substances follow the G protein coupled receptor pathway, and sweet substances follow the G protein coupled receptor pathway, this whole one that we went over. Okay, so out of this, it's not just glucose or sweet substances, it would also be bitter substances, and it would also be what else? We said umami substances. Okay, these would also work through this messenger system. Now there's another way that we could do this. Let's have another taste cell down here. Let's say here I have another taste cell. This one's a lot easier. And again, what would it be connected to? It would be connected to some type of nerve terminal here of some cranial nerve, and it would be taking this information away, right? What could be this mechanism? Let's start off with the salty. Now, on the membrane, this one's really, really easy. On the membrane, you have these channels that are open, and they're very specific to sodium chloride. Okay, but particularly that of sodium. Look what happens here. If this channel is normally open, sodium ions can rush into this cell. Sodium ions can rush into this cell. Now, normally, you're going to have this sodium ions flowing in and in and in. And then if you remember, based on that diagram that we've had before, if I have here, I show here I have a graph, and let's say here was resting membrane potential, and here's my threshold potential. When sodium keeps flowing in and in and in, he's bringing the resting membrane potential closer to threshold potential. Once he brings it to threshold potential, he opens up special voltage-gated calcium channels on this membrane. And calcium will start flowing into this cell. As calcium starts flowing into the cell, what's going to happen? Remember, what's inside of these actual taste cells? These little vesicles rich in certain types of neurotransmitters. What are those neurotransmitters? ATP and serotonin. Calcium is going to do what? It's going to activate the synaptoproteins on these guys to trigger them to bind with the cell membrane and release what chemicals? Serotonin and ATP, what will happen? It'll fuse with this actual membrane. And when it fuses with this membrane, what will happen? It'll release out the chemicals into this actual synaptic cleft. And when the serotonin and the ATP will stimulate these nerve terminals and activate action potentials that move down this cranial nerve. And why would the sodium move in? Because sodium is in really high concentration outside the taste cells, right? If you're having a salty substance. Now, one more. You know there's other channels here that are constantly usually going to be trying to be open. They're called potassium leakage channels, and potassium loves to leave the cell through this channel. Well, what if you have some type of sour substance? Let's say you have a sour substance. If you're having a sour substance, what is it going to be rich in? Protons. You know, protons, they see this channel, they're like, that's perfect for me. I'm going to go through this channel. So when the protons try to come through this channel at the same time that the potassium is trying to leave, guess what happens? the protons block the potassium ions from leaving the cell. So look, the protons come over here and they put like a plunger edge on this, and they're like, you ain't getting out, boy. And the potassium ions stay in here. If the potassium ions stay in here, what happens? The inside of the cell starts building up potassium ions, and the cell starts becoming very, very positive. As the cell starts becoming positive, what starts happening? It starts approaching from resting membrane potential to threshold potential. Once it reaches threshold potential, what happens? These calcium channels open, voltage-gated calcium channels open, and then calcium rushes in. When calcium rushes in, it triggers the release of serotonin and ATP onto these actual nerve terminals. All right. Thank goodness, that's done, right? So this was for sour, and this was for salty. All right. Now we're going to get to the good stuff, okay? Let me actually erase these words so that we can show these nerves now. Okay, now what we're going to have to do is we're going to see what of those cranial nerves that I mentioned, because I mentioned that these nerve terminals here are going to be carried on cranial nerves. First off, before we show which ones, which cranial nerves could it be? It could be cranial nerve 7. It could be cranial nerve 9. It could be cranial nerve 9, and it could be cranial nerve 10. Cranial nerve 7 is the facial nerve. Cranial nerve 9 is the glossopharyngeal nerve, and cranial nerve 10 is the vagus nerve. Okay. Now the facial nerve is going to be very specific. Let's do the facial nerve in this blue color right here. The facial nerve is going to be picking up taste. Any of those tastes, it doesn't matter, from the anterior 2/3 of the tongue. We're not going to be able to connect it to all of them, but you guys get the point. It's going to be picking up any type of taste sensations from what? From the anterior 2/3 of the tongue. Now the facial nerve is actually going to, we'll talk about this more when we talk about the trigeminal nerve, but it's actually going to run within a structure here, which is combined with other fibers, which is touch pain temperature fibers, which is coming from the trigeminal nerve here. Right? This is actually going to be moving through a structure here, and there's other fibers that actually run within this too. There's other fibers that actually go to a submandibular salivary gland, which is coming from the facial nerve also. This whole structure here is called the chorda tympani. Okay, and we'll talk about this more when we talk about the facial nerve. But what happens is these taste fibers are going to run with the chorda tympani. When they come, they're going to go to a specific type of nucleus, right? So these taste fibers are going to go to a specific type of nucleus that is going to be located within the pons, and it could be in the medulla too. And this nucleus, let's do this in this doo-doo brown color. This nucleus is going to extend, it could be from the medulla, and it could be in the pons. This nucleus is called the nucleus of tractus solitarius, right? The nucleus of tractus solitarius. And what happens is this facial nerve is going to give fibers to the nucleus of tractus solitarius, okay? And again, what is this cranial nerve? This is cranial nerve 7. So again, cranial nerve 7 is picking up taste sensations from the anterior 2/3 of the tongue, moving along with the trigeminal nerve, specifically the fibers that are going for touch pain temperature, and moving with, if I were to do it here in red, there is another nerve that actually runs in and out, and it goes to a ganglion, which we'll talk about, called the submandibular ganglion, which goes to the submandibular gland and the sublingual salivary gland. It comes and moves into the pons, and it goes to the medulla and supplies the nucleus of tractus solitarius. Sweet. Now let's do the next one. Let's do this one in a purple. Okay. Now if you remember, we had this circumvallate papilla from the posterior 1/3 of the tongue. And what else did we have? We had a little bit of fungiform papilla within the posterior 1/3 of the tongue. These guys are going to be picking up taste sensations from the posterior 1/3 of the tongue, from primarily the fungiform papilla and the circumvallate or vallate papilla. These are going to be carried on a special nerve here too. And this is called cranial nerve 9. Cranial nerve 9 is the glossopharyngeal nerve. Okay, now, the glossopharyngeal nerve will actually run through two different types of ganglia. You know, there's actually going to be a ganglia that'll actually run through. This ganglia is actually going to be called what's called the petrosal ganglion, or the inferior ganglion. That's where the cell bodies of these peripheral processes are coming, and then the central processes are going to move into the medulla. It actually run through a little hole called the, if you really want to know it, there's a hole here called the jugular foramen, and it moves through the jugular foramen here. And then it comes into the medulla, right? When it comes into the medulla, it supplies the, yeah, you guessed it, nucleus of tract solitarius, right? Okay. Then there's going to be, what else? Oh, and if you were, since we're being particular here with the ganglion, cranial nerve 7 will actually go to a specific ganglion located around the middle ear. It's actually called the geniculate ganglion. Geniculate ganglion. This can actually be affected during herpes zoster, like the shingles virus, okay? All right, sweet. Now we have this last part, the taste buds that are coming from the epiglottis, the taste buds that are coming from the pharynx. This is going to be cranial nerve 10, and cranial nerve 10 is going to be the vagus nerve. Let's do this bad boy in, let's do it in this blue here. Okay, so this guy here is going to be, not this blue, let's do it with this blue. All right, this is going to be the vagus nerve, and the vagus nerve is picking up, again, taste fibers from the, what? It's picking up taste fibers from the epiglottis, it's picking up taste fibers from the pharynx, and maybe even some small taste fibers from the soft palate, okay? But primarily from the epiglottis and a little bit from the pharynx. This nerve is actually going to run to a specific ganglion too, since we're mentioning them. And this ganglion that it comes to is actually called the nodose ganglion. Okay, the nodose ganglion, which is the inferior ganglion for the vagus. That will run, again, through what hole? It actually will run through a specific hole here, which is called the jugular foramen. And when it moves through the jugular foramen, it'll actually come into the medulla, and it'll go to the nucleus of tract solitarius. And since we're being picky here with all these darn holes here, this will actually go through the internal acoustic meatus. So internal acoustic meatus. All right. Now, these fibers, all these taste fibers, are going to come from the nucleus of tract solitarius, and these fibers are going to go upwards. When they go upwards, they give some branches off to a specific structure here. This structure that they drop off within the pons is a specialized nucleus area, and this area is called the pontine taste area. So it gives off branches to what's called the pontine taste area. Then, from the pons will send up fibers that go to a specific nucleus within inside of the thalamus in the diencephalon. This nucleus, if we were to draw the thalamus, let's say that we have the thalamus here. Say we draw the thalamus. Here's the thalamus. Let's say here we draw it like this. And it actually has the lamina right there. Inside of the thalamus, there's a specialized nucleus here that picks up this specific type of sensation. This nucleus is called the ventral posterior medial nucleus. Ventral posterior medial nucleus. So this ventral posterior medial nucleus is going to be receiving signals from the pontine taste area. Then the ventral posterior medial nucleus is going to send these fibers. You know, there's what's called the internal capsule. And then from the internal capsule will give out radiations to a specific part of the brain. You know what part of the brain it's actually going to be going to? It's going to the insula, which is going to be one of the lobes that's really, really deep to the actual temporal lobe. So it's going to be going to a specific part of the actual brain, which is going to be primarily that of the insula. And they call this, where these taste fibers are going to, they call this the primary gustatory cortex. But you got to be knowing that also from here, taste is not just going to be going to that, but it can also go to other areas of the brain too. Taste could actually go to the hippocampus so that we can store that memory of taste. It can also go to the hypothalamus, and the hypothalamus could also affect be affected by that. It could go to many different other areas too, right? It could even go to the amygdala. So taste could even go to the amygdala. Alrighty. Now, we've covered all of these different types of things. We've covered the pathway. And again, when these tastes get to the primary gustatory cortex, we'll have the conscious perception of that taste. One thing I do want to mention is, remember I told you that the olfactory pathways, so all the olfactory pathways, so let's say I put olfactory pathway, and these taste pathways are very intermingled. They're very intermingled, meaning that, again, I told you, taste, you need to write this down, taste is actually 80% smell. So without being able to actually have smell, if you have anosmia, taste is going to be pretty much absent also because they are very intermingled. So the concept of taste and smell is usually utilized as one concept also. There's very little diseases that we could really talk about that could actually cause some type of problem with taste. If you really were to think about it, it's usually related to, if there was the more common cause, obviously if you burn the taste buds, that's another thing. Do you know those taste cells? Those are actually epithelial cells. So if those are epithelial cells and you come into constant contact with hot or cold or foods that are causing friction, these suckers got to regenerate very quickly. These taste cells actually regenerate every 7 to 10 days. Whereas within the olfactory, remember the olfactory one? That one has a little bit longer. That one's about 6 to 8 weeks. So the olfactory actually nerves regenerate every 6 to 8 weeks. The taste cells, they regenerate every 7 to 10 days because they're in constant contact with harsh environments. But if there was any type of situation which there was damage to the facial nerve, which can happen in Bell's palsy, if there's some type of damage to the actual glossopharyngeal nerve due to some type of tumor that could be developing in some part, or maybe even multiple sclerosis or certain types of medullary infarcts, some type of disease of the vagus nerve, then yeah, you might lose taste to the actual certain parts of the tongue. All right, but we're not going to spend too much time talking about that. All right, engineers, in this video we talked about a lot of information about gustation or taste. I hope it all made sense. I hope you guys really did enjoy it. If you guys did, please hit the like button, comment down in the comment section, and please subscribe. All right, engineers, as always, until next time.